So this is the in-class activity for Chapter 16, the autonomic nervous system and higher order functions. So Part 1, insecticide poisoning. Sophia is a 44-year-old woman who had spent much of the day working in her garden. A blustery wind caused her to involuntarily inhale the insecticide that she was spraying throughout the garden. When she began wheezing severely, she was taken to the emergency room by her significant other. The attending physician observed other symptoms, including constricted pupils, a slowed heart rate, and excessive salivation. The patient was treated with an IV of atropine. Number 1. A lot of insecticides contain organophosphates, which can inhibit acetylcholinesterase. What is the function of acetylcholinesterase? So if you remember from back in Chapter 10, acetylcholinesterase is an enzyme that breaks down acetylcholine in the synapse, which limits the actions of acetylcholine as a neurotransmitter. Number 2, which types of autonomic receptors are excessively stimulated as a result of the inhibition of acetylcholinesterase? So there are 2 types of acetylcholine receptors in the autonomic nervous system. There are nicotinic receptors, which are found in both the sympathetic and parasympathetic divisions. These are ionotropic receptors, meaning that they directly open ion channels when the acetylcholine binds, and this is going to allow sodium to flow into the postsynaptic membrane, which results in excitation or depolarization of the target cell. The other type of acetylcholine receptor is the muscarinic receptor. These are found only in the parasympathetic division, and these are metabotropic receptors, meaning that the neurotransmitter binds to a G protein, and there is activation of a second messenger cascade, and down the stream, later down the signaling pathway, you may get the opening of ion channels. And in the case of the muscarinic receptors, these can be either excitatory or inhibitory, depending on the receptor subtype. So there are actually 5 different subtypes of muscarinic receptors. So the bottom line is that without acetylcholinesterase to break down acetylcholine, both of these types of receptors would become overstimulated. Question number 3. Which division of the autonomic nervous system would be affected the most and why? So let's review the 2 autonomic pathways and the neurotransmitters and receptors that they use. So if we start with the sympathetic division, we have our preganglionic neuron, which leaves the spinal cord and travels out to a peripheral ganglion. So here's the autonomic ganglion, and there it's going to form a synapse with the ganglionic neuron, whose axon is called a postganglionic axon or a postganglionic fiber. And this neuron is going to go further out into the periphery, where it's going to form a synapse with a peripheral effector, which could be cardiac muscle, smooth muscle, or some type of gland. Our parasympathetic division has the same similar setup. We have a preganglionic neuron, which leaves the spinal cord or brainstem and goes out to an autonomic ganglion, where it forms a synapse with a ganglionic neuron, which also has a postganglionic axon, which is going to go further out and also communicate with a peripheral effector, which would be cardiac or smooth muscle or a gland. So at this first synapse that takes place inside the autonomic ganglion, the neurotransmitter is acetylcholine, and the receptors on the postsynaptic membrane are nicotinic receptors. And so this is true for both the sympathetic division and the parasympathetic division. However, at the second synapse, the one between the postganglionic neuron and the peripheral effector, we have a difference. In the sympathetic division, the postganglionic synapse uses the neurotransmitter norepinephrine, and the receptors on the postsynaptic membrane of the peripheral effector are going to be alpha and beta receptors. In the parasympathetic division, at the postganglionic synapse, we're going to use acetylcholine again, but this time the receptors that are going to be on the postsynaptic membrane are muscarinic receptors. So both divisions use acetylcholine at nicotinic receptors at their first synapse, but only the parasympathetic division uses acetylcholine a second time at the second synapse. So because the parasympathetic division uses acetylcholine at both the preganglionic and postganglionic synapses, it would be the most affected by organophosphate insecticides, which block acetylcholinesterase. Also note that muscarinic receptors are activated at lower concentrations of acetylcholine than nicotinic receptors, which makes them more susceptible to having too much acetylcholine around, which is the result of blocking acetylcholinesterase. So the muscarinic receptors are going to be affected earlier and quicker than the nicotinic receptors. So number 4, under what conditions does this division of the autonomic nervous system normally predominate? So remember, on the previous question, we determined that it was the parasympathetic division that is the one that's going to be the most affected by this type of insecticide. And the parasympathetic division of the autonomic nervous system predominates under resting or baseline conditions. And this system is often called the rest and digest division. In contrast, the sympathetic division is usually only active during periods of exertion, stress, or emergency, and it's often called the fight or flight division. So these two systems typically have opposing functions. You can sometimes think of the sympathetic system as being the gas pedal and the parasympathetic system as being the brake pedal. Number 5, explain how the insecticide could have caused her symptoms. Include a description of the pathway that information flows in the division most affected. So again, we are looking at the parasympathetic division, which would be the most affected by organophosphate insecticides. So let's start by looking at the pathway first. So the pathway starts in the autonomic control center of the hypothalamus. So I start with a neuron that starts off in the hypothalamus. And this neuron is going to run down to the brainstem or the spinal cord, specifically in the areas of cranial nerve 3, 7, 9, or 10 within the brainstem, or S2 to S4 within the spinal cord. And in those two areas, the hypothalamic neuron is going to synapse onto what is called a preganglionic neuron. So again, the cell body of this preganglionic neuron in the parasympathetic division is either going to be in the brainstem at the location of cranial nerve 3, 7, 9, or 10, or in the spinal cord segments S2 to S4. Next, the preganglionic axon is going to leave the brainstem or spinal cord, and it's going to travel to the autonomic ganglion. And this is a long preganglionic axon. And again, remember, when you see axon, that can be interchangeable with the word fiber. So the autonomic ganglion in the parasympathetic division is going to be either close to the target effector, and we call those terminal ganglia, or it's going to be inside the target effector, and we call those intramural ganglion. So the ganglia are near or within the target organs and effectors. Then the preganglionic neuron is going to synapse onto a neuronal cell body in the ganglion. This is called the ganglionic neuron. And at this synapse within the ganglion, we're going to use acetylcholine as the neurotransmitter, and there's going to be nicotinic receptors on the postsynaptic membrane. The postganglionic axon is then going to leave the ganglion and travel a short distance to the target organ or effector. And notice that this is a short postganglionic axon because the ganglion is already located either near or within the target. So a long preganglionic axon and a short postganglionic axon. And then finally, at this synapse, we're going to have acetylcholine as the neurotransmitter, and the muscarinic receptors are going to be on the postsynaptic membrane. So that's the pathway that information flows in the parasympathetic division. So now let's look at how this insecticide could have caused her symptoms. So as a reminder, we're looking at the parasympathetic division. Also, as a reminder, remember that the muscarinic receptors that you find in the target organs can be either excitatory or inhibitory. So she had severe wheezing because of excitation, which led to contraction of the smooth muscles in her bronchioles in the lungs, and this resulted in narrowed airways. So it was hard for her to breathe in because of the constriction of the airways, and that causes wheezing. Her constricted pupils occurred because of the excitation of the pupillary constrictor muscles in the eye. She had a slowed heart rate called bradycardia because of inhibition of pacemaker cells in the heart. And she had excessive salivation because of excitation of the salivary glands. Don't get too bogged down in these details. The take-home message for this question is to know what the functions of the parasympathetic division are. So the parasympathetic division is responsible for constriction of pupils, a decreased metabolic rate, a decreased heart rate and blood pressure, constriction of respiratory pathways, increased secretion by salivary and digestive glands, increased motility and blood flow to the digestive tract, increase in absorption of nutrients by cells, inhibition of glucose release into the blood, so we want to take the nutrients out of the blood and bring them into the cells, stimulation of urination and defecation, and sexual arousal and erection. Number 6, atropine blocks muscarinic receptors. How would this have helped the patient? So the initial symptoms of mild organophosphate insecticide poisoning all involve the parasympathetic division with the exception of sweating. So you have increased saliva and tear production, you have small pupils, you have wheezing, a slow heart rate, vomiting and diarrhea, and sometimes involuntary urination. All of these involve the parasympathetic division. The sweating is the only symptom in this initial list that actually involves sympathetic activation, and that would have been because of the nicotinic receptors along the sympathetic pathway. And since the cause of the majority of these symptoms, especially the most dangerous ones like the slow heart rate and the wheezing, is the action of acetylcholine at muscarinic receptors, one of the first responses to organophosphate poisoning is to give the patient atropine. So the atropine blocks the muscarinic receptors, and that stops the parasympathetic postganglionic neurons from being able to exert their effects on the peripheral effectors. So even though the acetylcholine is still around, the atropine gets in their way so that it can't bind to the muscarinic receptors. And so this sort of addresses the life-threatening part, especially the wheezing and the slow heart rate. But note that as the dosage of the insecticide exposure increases, you can also get activation of the sympathetic nervous system because of activation at the nicotinic receptors. So you can also, with higher doses, see anxiety, convulsions. The slow heart rate can swap to elevated heart rate, and you can end up with hypertension, which is high blood pressure. So depending on the dose of poisoning, a second drug may be given after the atropine, which actually reverses the inhibition of the acetylcholinesterase by the organophosphate insecticide. But the atropine is given first to stop the more life-threatening immediate symptoms that appear. Number 7, if exposed to high enough doses, what effect might the insecticide have on the patient's skeletal muscles? With a high enough dose, the nicotinic receptors in the neuromuscular junctions, or NMJs, become affected. And so overstimulation by acetylcholine at the neuromuscular junction can result in muscle tremors, fasciculations, which is that name for the twitch of a whole motor unit, and myoclonic jerks, which are quick, jerky movements, typically of the limbs. And so when acetylcholine sticks around in the neuromuscular junction, because remember, the insecticide is blocking acetylcholinesterase, so acetylcholine is going to stick around for too long. And so it's going to activate the nicotinic receptors on the muscle cell, but then the muscle cell is going to have an action potential and it's going to be excited, but then it's not going to be able to repolarize after that initial depolarization. So after you have these initial muscle symptoms from the rounds of muscle contractions, you'll then fall into a state of paralysis where the muscles cannot contract a second time. Number 8, sarin is an extremely toxic organophosphate and is one of the most toxic of the known chemical warfare agents. It is classified as a weapon of mass destruction. What is the likely cause of death resulting from exposure to sarin? So the root cause of death from sarin exposure is actually asphyxia, which is the loss of oxygen, and this occurs from respiratory failure. And the reason you have respiratory failure is a combination of the constricted airways that we talked about before, and then as the dose increases and affects muscles, you have spasming and then paralysis of the breathing muscles. So the bottom line is that you end up dying from a lack of oxygen because you can't breathe. Sarin is rapidly fatal. Death can occur within 1 to 10 minutes after inhalation of a fatal dose. And unfortunately, it's not a quick or pleasant death because the person also suffers from all of the other effects that we just previously mentioned that occur with this acetylcholinesterase blocker. So they may have vomiting, defecation, urination, convulsions, drooling, all of that before they have the final stage of asphyxia where they can't breathe and die. And because sarin is such a nasty drug, it was outlawed by the Chemical Weapons Convention in 1997. Part 2, additional autonomic questions. Number 1, you are working diligently at your desk studying for your A&P final exam when your friend comes up quietly behind you and says, "Boo!" You jump and your heart starts pounding rapidly. What division of the nervous system was activated? Explain the pathway of electrical impulses beginning with the brain and ending with the heart. In addition to the altered heart rate, what else is going on? So let's start off by looking at the pathway. So we're going to look at the pathway of information. And again, we are focused on the sympathetic division this time. We looked at the parasympathetic division last time. Now we're going to focus at the sympathetic division, which is the one that would be activated if you're frightened. So again, we're going to start in the autonomic control center of the hypothalamus with the hypothalamic neuron. Again, we are going to run from the hypothalamus down to the spinal cord where we are going to synapse onto a preganglionic neuron. The cell body of the preganglionic neuron in the sympathetic division is going to be in the spinal cord between spinal segments T1 and L2. Then the preganglionic axon is going to leave the spinal cord and travel to the autonomic ganglia. And this is going to be a short preganglionic axon. And the reason this one's going to be short is because the autonomic ganglion is close to the spinal cord. So because the ganglia is close to the spinal cord, this axon doesn't have to travel very far. So there are two major types. There are chain ganglia, which are to the sides, each to the left and right side of the spinal cord, and 3 collateral ganglia, which are just anterior to the spinal cord. The one exception in this case is the adrenal medullae, which is a modified ganglion that also is innervated by the sympathetic division. So the preganglionic axon is going to synapse onto this neuronal cell body in the ganglion. And this synapse, just like with the other division, is going to release acetylcholine, and you're going to have nicotinic receptors on the postsynaptic membrane. The postganglionic axon is going to leave the ganglion, and in this time, it's going to travel a long distance to the target effector. And that's because the ganglion is close to the spinal cord, the target effector is out in the periphery. So now we have a long postganglionic axon. And then this final synapse between the postganglionic axon and the effector is going to be mostly norepinephrine. And then you're going to have alpha and beta receptors on the postsynaptic membrane. So that is the pathway of information in the sympathetic division. And so if we look at what caused the altered heart rate, let's first remember that in the sympathetic division, we have 2 types of receptors, alpha and beta. Alpha 1 is an excitatory receptor. Alpha 2 is an inhibitory receptor. Beta 1 is excitatory, beta 2 is inhibitory, and then beta 3 is only found in adipose tissue. So the elevated heart rate that you feel after you've been scared is because of activation of beta 1 and alpha 1 receptors in the heart, which causes an increase in the heart rate and the force of contraction of the heart. But again, the take-home message is to think about what the sympathetic division is doing. So the sympathetic division is responsible for heightened mental awareness, dilation of the pupils, an increased metabolic rate, an increased heart rate and blood pressure, dilation of the respiratory passages, an increased respiratory rate, reduced digestive and urinary functions, activation of energy reserves, so taking both fat and glucose out of their stores and putting them into the blood, activation of the sweat glands, and orgasm. Number 2, what are some key similarities between the sympathetic and parasympathetic nervous systems? What are some key differences? So let's start with the similarities. Both systems are controlled by the hypothalamus. So control over both systems starts with the autonomic control centers within the hypothalamus. Both systems have preganglionic neurons that originate in the brainstem or spinal cord. Both systems have autonomic ganglia, which are places in the periphery where you have neuron cell bodies. Both systems have ganglionic neurons, so the cell bodies are in those ganglia. And then the ganglionic neurons have postganglionic axons that go out to peripheral effectors. Both systems also use cholinergic synapses between the preganglionic axon and the ganglionic neuron, so they both release acetylcholine at that synapse. And both systems use nicotinic receptors at that cholinergic synapse. So the postsynaptic membrane will have nicotinic receptors on it. So those are the key similarities. Now let's take a look at the key differences. The two systems have different locations of the preganglionic cell bodies in the central nervous system. In the sympathetic division, the preganglionic cell bodies are between T1 and L2 spinal segments. In this parasympathetic system, the cell bodies are where cranial nerve 3, 7, 9, and 10 originate in the brainstem, and then spinal segments S2 to S4. They also have different preganglionic axon lengths. The preganglionic axon is short in the sympathetic division and long in the parasympathetic division. They also have different ganglia location. In the sympathetic division, the ganglia are close to the spinal cord, that is the chain ganglia and the collateral ganglia. In the parasympathetic division, the ganglia are close to or within the target organ, and that is the terminal ganglia and the intramural ganglia. They also have different postganglionic axon lengths. In the sympathetic division, the postganglionic axon is long, and in the parasympathetic division, the postganglionic axon is short, so opposite to the preganglionic axons. Different neurotransmitters are used by the postganglionic axon. So in the sympathetic division, it's mostly norepinephrine that's used at that second synapse, and norepinephrine is going to bind to alpha and beta receptors. And in the parasympathetic division, it is acetylcholine used at that second synapse, but this time it's binding to muscarinic receptors. They also have a different range of effects. So in the sympathetic division, the range of effects of one postganglionic axon is broad. So because the postganglionic axons are long, they can branch repeatedly, and so one axon can go out and innervate multiple targets. Whereas in the parasympathetic division, the targets are very specific. So again, the postganglionic axon is short. Sometimes it's already inside the target organ, so it's not going to branch out and go to different targets, so you have one axon per target. And then finally, they have different functions. You can sum up the sympathetic division functions by saying fight or flight, basically preparing for stress or an emergency. And the parasympathetic division dominates during normal conditions, and we call it the rest and digest. Part 3, higher order brain functions. Number 1, describe how memory consolidation occurs. What structures are required? What are some of the events that take place on a cellular level? So remember that memory consolidation is the process of converting a short-term memory into a long-term memory. Short-term memories are stored in the hippocampus, and then they must be transferred to the cerebral cortex in order to have them in your long-term storage. So we know that the hippocampus and the amygdala are both absolutely required for memory consolidation to occur. If you remove these structures, you impair the ability to form new memories, like we saw with the patient HM. We also know that the nucleus basalis plays an important role in memory. It's not yet fully understood, but we do know this is the first area of the brain that degenerates in Alzheimer's disease. So the cellular changes that take place. So remember that the term memory engram is the term we use to describe the specific connections between neurons that represents a specific memory. Right, so this is all about the connections and the synapses between two or more neurons. So as a particular memory is strengthened, there is increased neurotransmitter release at these synapses. You get more receptors added to the postsynaptic membrane for the neurotransmitter to bind to, and both of these things together is going to give you stronger synapses. You're also going to get the formation of new synapses between the neurons involved in the engram. And the involved neurons are also going to grow new axon terminals and new dendrites to further strengthen their link. So the more of these cellular changes that you have that take place, the stronger these particular neurons are linked together, and thus the stronger the memory. And always remember, repetition and sleep are essential for these cellular changes to take place. You have to repeat information over and over again in order to tell the brain that it's important so that it will undergo these cellular changes, and then these cellular changes only happen while you are sleeping. Number 2, think back to your last big celebratory event. It could be a birthday, a graduation, a marriage, a new birth, etc. Describe how various aspects of this memory are stored in the cortex. So there's a lot of possible answers to this question. You could have had many different answers. I'm going to use a birthday party as an example so you can kind of see the kind of answers I was looking for. So you do have visual memories that are stored in the visual areas of the brain, and these are the pictures that you see in your head when you're thinking back to a particular memory, like the decorations in the room, the way the cake was decorated, the costumes people were wearing, etc., the color of the gift paper. Auditory areas are going to store auditory memories, like the sound of the music that was playing, the sound of those party favors that make that little squeaking noise, the sound of everybody talking, etc. Everybody singing happy birthday off key, those types of memories. The frontal lobe is where you're going to store factual memories. So how many people showed up to your party, who showed up, the name of each gift you got, what number birthday it was. So it was your 18th birthday, 20th birthday, 30th birthday, etc. Your sensory areas will store sensory memories. So like, for example, one that I always remember, how the rubber band on the birthday hat, pinched your neck, because it's never very comfortable to wear those little birthday hats. You'll also, I didn't put it on here, but you also could have like gustatory areas that remember gustatory memories, like what the cake tasted like. And then you have limbic areas that store emotional memories, like how much fun you had, the enjoyment, or maybe you hated it, the emotions that went along with that event. Number 3, describe the 3 hypotheses for why sleep is vital for life. The first one is that we sleep to forget. So irrelevant memories, those synapses that are not used much, are trimmed away and reduced to make the brain more efficient in terms of energy use. The second one is that we sleep to remember. So important memories, the synapses that you use frequently, are strengthened, and this is that process of memory consolidation. and you actually have the cellular processes that strengthen those synapses that we talked about in question 1. And then we also sleep to clean our brains. So recent research shows that the flow of CSF, cerebrospinal fluid, increases while we sleep, and built up waste products and proteins are washed out of the brain during sleep. And so, like I mentioned in the lecture, it's more likely that all 3 of these things contribute to why we sleep. And it's thought that death could possibly result from the lack of sleep, because I mentioned in the lecture, if you don't sleep, you will eventually die. It is fatal. And it's thought that this probably occurs mainly because of number 1 and number 3. You end up building up too many temporary synapses that you don't remove while you sleep, and that starts causing overwhelming energy demands on the brain. And then there can also be a buildup of toxic waste products from the lack of the increased CSF flow, and both of these could eventually lead to the brain shutting down. Number 4, describe the system in your brain that is responsible for maintaining your awake state. So arousal or wakefulness is maintained by the reticular activating system, or RAS, and this is the superiormost part of the reticular formation that spans the brainstem. So remember, the reticular formation spans throughout the medulla oblongata, the pons in the midbrain, and then the RAS sits at the very top in the midbrain. So the RAS sends signals out to the thalamus and the cortex to keep us awake. Anything that activates the RAS while you're sleeping can wake you up when you're sleeping. And I mentioned way back in chapter 14 that the RAS can also learn what stimuli to ignore. So when you first move to a new house, for example, and a bunch of different noises wake you up at night, and the RAS eventually learns which noises it can ignore and which noises that it's going to wake you up for. And then the longer that we are awake during the day, the more the activity in the RAS decreases, which then makes us feel lethargic and tired and compelled to sleep. And as I also mentioned in the lecture, the preoptic area of the hypothalamus also participates in the sleep-wake switch that allows us to move between states. So when the reticular activating system and other arousal symptoms are on, the preoptic area is off, and vice versa. When the preoptic area is on, the RAS is off. And number 5, what neurotransmitter is associated with addiction? Why do drugs that affect this neurotransmitter cause addiction? So dopamine is associated with addiction because of its role in the reward pathway. Dopaminergic neurons from the ventral tegmental area in the brainstem play a key role in the feelings of pleasure we get because of the reward pathway. So anytime you feel good about anything, a job well done, you're eating your favorite cake, having sex, having fun, telling jokes, whatever gives you a feeling of pleasure, it's because of the dopaminergic neurons that are playing a role in this reward pathway. So the reason that drugs become addictive is because in a lot of these cases, the drug causes excessive dopamine release. So that's why it feels great the first time, right? That's what causes the high. But in response to that excessive dopamine release, the body starts reducing the number of dopamine receptors available. So now there's fewer receptors for the dopamine to bind to. And this eventually leads that even normal things that you used to take pleasure in, like your favorite flavor ice cream or, a nice walk in the woods, whatever you typically, your little small daily pleasures no longer feel pleasurable. So you actually feel worse when you're not taking the drug. You no longer feel normal. You're like depressed all the time. You don't find pleasure in anything. And eventually you get to the state where you have to take the drug just to feel normal. And again, that's because of the reduced dopamine receptors. And then you have to start taking more and more drug to try to get the high that you got the first time. And that's the process of tolerance, which can lead to a deadly cycle. And that is the end of chapter 16.